Hello everyone. In this project, we propose a complementary test for white-eye flies to explore how different genetic mutations contribute to the white-eye phenotype in Drosophila. A complementary test determines whether mutations causing the same phenotype are on the same or different genes. If they fail to complement, the phenotype persists, indicating they're on the same gene. If they complement, the wild-type phenotype returns, confirming they're on different genes. Drosophila, commonly known as the fruit fly, is a widely used model organism in genetics due to its many advantages. Its simple chromosome structure, comprising 3 autosomes and 1 X chromosome, makes it ideal for studying inheritance. Additionally, Drosophila displays a variety of eye colors, including white-eye mutations, the focus of this project, which can result from mutations in different genes. The white-eyed phenotype in Drosophila can arise from 2 types of mutations. It may result from an autosomal double mutation, the Princeton mutation, which causes a brown eye phenotype, and the Harvard mutation, which causes scarlet eye phenotype. When these 2 mutations occur together, a phenomenon called epistasis leads to the white eye phenotype. Alternatively, the white eye phenotype can be caused by a sex-linked mutation located on the X chromosome. Based on our current knowledge, our study relies on 3 main assumptions, or more precisely, known facts. First, there are 2 strains of white-eye flies, one with a sex-linked mutation and another with a Princeton-Harvard double mutation. Second, one mutation is sex-linked, while the other is autosomal. Lastly, the white-eye phenotype is recessive, appearing only in individuals with 2 mutated alleles. These assumptions guided our following experimental design and expected results. Since one strain of our white-eye flies has a sex-linked mutation, we propose a set of reciprocal crosses. This method involves swapping the sexes of the parent flies in 2 genetic crosses to examine the sex linkage of specific genes. In cross 1 that we proposed, male flies carrying the sex-linked mutation are crossed with female flies with autosomal double mutations. In cross 2, male flies with autosomal mutations are crossed with female flies carrying the sex-linked mutation. So, what are the expected results based on our current knowledge? For cross 1, we expected 100% of the females to be wild-type and 100% of the males to also be wild-type, as shown in the Punnett square here. This indicates that cross 1 will show 100% of complementation. However, for cross 2, due to the sex-linked mutation inherited from the female parent, all the males would have white eyes, while the females would remain 100% wild-type. In this case, we expect half the progeny to display complementation.
In genetic research, our initial assumptions are always subject to scrutiny. We have 3 critical assumptions we need to carefully examine. First, we examine that we have 2 distinct strains of white-eyed flies, one with a sex-linked mutation and another with a Princeton and Harvard double mutation. Let's consider a scenario where we actually only have 1 strain of white-eyed flies. If our white-eyed parents have only the sex-linked mutation, both crosses would yield identical results. We see 100% white-eyed offspring in both female and male progeny, and this would suggest that we're dealing with a single mutation. Similarly, if the white-eyed flies possess only the Princeton-Harvard double mutation, we would again observe 100% white-eyed offspring in both crosses. This is to emphasize our initial hypothesis of having 2 separate mutation strains. Second, we believe that the sex-linked mutation is on the X chromosome, while the Princeton and Harvard mutations are autosomal. If both the white-eyed mutations are autosomal, our crosses would produce 100% wild-type offspring. This specific outcome would indicate that the mutations are located on different genes. And the genetic complexity here lies in understanding how these autosomal mutations interact and potentially complement each other. Conversely, if mutations are sex-linked, we observe a different pattern. 100% wild-type females and 100% wild-type males in both crosses. This scenario highlights the intricate nature of sex-linked inheritance and how chromosomal location can dramatically influence gene expression. Third, While working under the assumption that the white-eyed phenotype is recessive, meaning only homozygous individuals will express the trait, we have to look at the nature of the white-eyed mutation. These are typically loss-of-function mutations. The complexity of these mutations would make it extremely unlikely that they would be dominant.
Now, let's sum up the possible outcomes of a reciprocal crosses, which can be categorized into 4 key scenarios. First, fail to complement. If both crosses produce 100% of white-eyed progenies, this indicates that there are no 2 strains of white-eyed flies, and the mutations occur on the same gene. In this case, the phenotype persists because the mutation failed to complement each other. Second, complete complement. If both crosses produce 100% of wild-type progenies, this suggests that we do have 2 strains of white-eyed flies, and both mutations are autosomal, fully complementing each other. Third, half complementation. In other words, half of the progeny of both crosses show complementation. If both crosses result in 100% of wild-type females and 100% of white-eyed males, this indicates that both types of white-eyed mutations are sex-linked. Finally, partial complementation. In this scenario, cross 1 produces 100% of wild-type progenies, while cross 2 results in wild-type females and white-eyed males. This scenario only occurs when there are 2 strains of white-eyed flies, with one mutation being autosomal and the other sex-linked. Based on our current knowledge, we believe this last scenario reflects the actual results of our experiment.
Our complementary tests serve 2 primary purposes. First, we aim to confirm whether we are indeed working with 2 distinct white-eyed mutation strains. The genetic crosses will help us distinguish between single and multiple mutation scenarios. Second, we seek to determine the chromosomal location of these mutations. Are they truly a combination of sex-linked and autosomal mutations, or do they share the same chromosomal origin? By challenging our initial assumptions and analyzing the cross results, we can reveal the genetics behind the white-eyed fruit flies.